Welcome back, everyone. Today we are going to learn about the integument of an insect. The integument of insects, like that of other animals, is an outer dermoskeletal covering of the body and is derived from the embryonic ectoderm. It is functionally a composite structure that serves as a skin, skeleton, food reservoir, and switches on post embryonic development by undergoing molting periodically. So, when we learn about the structure and chemistry of this insect integument, we could see different structures like the basement membrane, the epidermis, cuticle, along with some other structures like the epicuticle, the cement layer, the wax layer, the polyphenol layer, cuticulin layer, the chitinous cuticle, and we'll be learning some specific things about this like the lamellar organization, the pore channels, then chitin protein microfibrils, chitin, biosynthesis of chitin, how chitin is formed, then chitin microfibrils, beta chitin, alpha chitin, gamma chitin, then different models of this chitin's microfibril arrangement like helicoidal model, screw carpet model, Blackwell and Weiss model, etc. And some important proteins involved in this like anthropodin, resilin, the sclerotin, lime pigments, and some different cuticular modifications like microtrachea, clothing hairs, glandular hairs, sensory hairs, the wing scales, which can be considered as a modification of this cuticular structure, the wing scales, spines, microtrachea, apodemes and apophyses, cuticular membranes, along with ecdysial lines. Then we will be learning about an important phenomenon of this molting, which is very important in case of an insect. What is that molting and how molting takes place? All those things. Then sclerotization, next important topic, sclerotization. All these topics comes under this integument portion. So we will be learning about all these one by one. If I can complete all these portions in this video, that's okay. If I can't, I will split this whole chapter into different videos. Okay, so first let's learn about the integument. The integument of insects, like that of other animals, is an outer dermoskeletal covering of the body and is derived from the embryonic ectoderm we mentioned earlier. It is functionally a composite structure that serves as a skin, skeleton, food reservoir, and switches on post embryonic development by undergoing molting periodically. Structure and chemistry. It is basically composed of three layers: the inner basement membrane, middle epidermis, and the outer cuticle. So, three layers are there. Inner basement membrane is there, middle epidermis is there, and outer cuticular layer. The epidermis is commonly called as the hypodermis as it lies below the cuticle. It is the only layer of integument that is cellular, while the inner basement membrane as well as the outer cuticle both are acellular structures. The epidermis secretes the cuticle, and the cuticle is modified later onto different skeletal sclerites, appendages, sensory organs, and internal lining of the fore and hindgut. So, three layers are there. It is the only layer of integument that is cellular. Which one? Epidermis. While the inner basement membrane as well as the outer cuticle both are acellular structures. The epidermis sclerites secretes the epidermis, secretes the cuticle, and the cuticle is modified later on to various skeletal sclerites, appendages, secretory organs, sensory organs, and internal lining of the fore and hindgut. The tracheal system, some organs of reproduction, and exocrine glands. All these modifications are created. So first let's learn about the basement membrane in detail. It is formed from the degenerated epidermal cells and appears as a non-living, amorphous, granular inner lining of the integument. It separates the epidermis from the hemocoel. It is about zero point 0.5 micron thick. The histochemical studies reveal that it is composed primarily of the neural mucopolysaccharides. I'm sorry, neutral mucopolysaccharides, not neural. Neutral, that was a mistake. Neutral mucopolysaccharides. On the inner surface of the basement membrane are attached to the muscles, hemocytes, and inocytes. Sometimes the stellate tracheal cells, collagen fibrils, and connective tissue fibrils are embedded in the basement membrane. And next comes the epidermis. It is an unicellular layer formed from the polygonal cells. The polygonal cells may modify into cuboidal or columnar cells having indistinct cell boundaries at this time of molting. However, in the old adult insects, the epidermal cells undergo degeneration and the epidermis is represented merely by nuclei lying beneath the cuticle. During post-embryonic development, the epidermal cells are well defined. The nuclei are polyploid, possessing a large number of nucleoli. The cytoplasm is characterized by containing various kinds of pigment granules. The adjacent epidermal cells are held with one another by means of cytoplasmic process, the desmosomes. Each epidermal cell produces a large number of cytoplasmic processes apically. The pore canals traversing the cuticle and opening above a cuticulin layer. The epidermal cells differentiate in some regions of the integument and constitute various types of mechano and chemoreceptor organs. The dermal glands, and particularly in dipteran larvae, the peristigmatic glands around the spiracles. Each dermal gland is formed by a group of 3 cells. The medial cell constituting the body of the gland and termed as the trichogen cell, while the remaining cells constitute the duct of the gland and are termed as tormogen cells. During molting, some epidermal cells modify into the so-called molting glands, which secrete the molting fluid that digests the old endocuticle to ensure further growth of the new cuticle. Sometimes the myofibrils penetrate the epidermis from the site of muscle attachment. Then about the next layer, that is a cuticle. The cuticle is a secretory product of the epidermis. It forms an outermost thick layer of the integument and determines surface patterns and physicochemical properties of integument. It is differentiated into 3 major regions like outer epicuticle, middle exocuticle, and inner endocuticle. The epicuticle is non-chitinous while the exo and endocuticles are chitinous regions. The exo and endocuticles are differentiated from the initially secreted procuticle due to sclerotization or tanning that take place in the upper region. The exocuticle therefore represents the sclerotized or tanned, and the endocuticle non-sclerotized or untanned, undifferentiated regions of the cuticle. Besides these 3 static regions, 2 more non-static regions develop at the time of molting between the exo and endocuticle as a transitional semi-hard and little darkened region commonly known as a mesocuticle. The outer region arises in between the endocuticle and epidermis in the form of a membrane and known as ecdysial membrane or the subcuticle. These structures can be seen only during larval and pupal molts and are completely absent from the cuticle of adult insects. The functional significance of these regions is still obscure. Then epicuticle is there. It is very thin outermost layer and varies in thickness from 0.03 to 0.4 micron. It is composed of 3 or 4 superimposed layers: outer cement layer, second wax layer, third polyphenol layer, and inner one cuticle. The polyphenol layer is reported only in some insects. So first, the epicuticle layer is there, which has some subparts, not substructures, like the cement layer. It is secreted by the dermal glands or so-called Wersch's gland in Lepidoptera. It is composed of lipoprotein complex resembling the natural product shellac secreted by the lac insect, Lactifer laca. It also contains the carbohydrate like a lactose. It functions as a varnish and provides protective external surface to the integument. It absorbs the mobile lipids that are used for sealing over the surface aberrations in order to prevent water loss from the body. It serves as a reservoir for lipids too. And the wax layer is there. It is a prominent layer about 0.25 micron thick, containing partially oriented wax molecules. It is composed of hydrocarbons 48 to 58%, free fatty acids 15 to 18%, esters 9 to 11%, cholesterol 2 to 3%, and polymers 12 to 15%. It does not contain alcohols, unlike that of plant waxes. It simply consists of long chain hydrocarbons and esters of both fatty acids and alcohols. It is a monolayer either in solid or liquid phase, and hydrophilic groups of the molecules are absorbed on the cuticle layer. It serves as a waterproof layer of the integument, as water molecules cannot pass through closely packed wax molecules. And next comes the polyphenol layer. This layer is described in the blood-sucking bug Rhodnis prolixus and appears often in the liquid state as a non-static layer containing various types of polyhydric phenols like homocatechol, protocatechol, propacatechol, hydroquinone, pyrogalol, etc. The polyphenols are transported from the epidermal cells through the pore canals and accumulate on outer surface of the cuticle layer. In some insects, in spite of forming an independent layer, polyphenols are used to impregnate the cuticle. At the time of sclerotization, these polyphenols are oxidized to quinones on the presence of an enzyme. The phenol oxidase and then the quinones tan first the proteins of cuticle layer and later proteins outer cuticle to produce the exocuticle. Then next the cuticle layer. It is a refractile amber colored layer consisting of lipoprotein, the cuticle. It is highly resistant to the mineral acids and most of the organic solvents. It serves as a permeability and growth carrier and determines the surface properties of the integument. And next comes the chitinous cuticle, exo and endocuticle. So we mentioned the different layers of epicuticle like the cement layer, wax layer is there, and polyphenol layer. Once again, I'm going to recap all this. The basement membrane is there, epidermis, cuticle. And cuticle has one layer called the epicuticle, which is further divided into three layers like cement layer, wax layer, polyphenol layer. Cement layer, wax layer, polyphenol layer, and the cuticulin layer. One more layer is the cuticulin layer. The chitinous cuticle, exo and endocuticle, it is a lamellar. Wait a minute. Epicuticle is further divided into four layers. Cement layer, wax layer, polyphenol layer, cuticulin layer. Okay. In the chitinous cuticle, exo and endocuticle, it is a lamellar or stratified structure differentiated as an outer exocuticle and inner endocuticle. The exocuticle is darkly pigmented, hard and sclerotized, from which are differentiating secondarily the sclerites in different regions of the body. Due to tough and inelastic properties, the exocuticle provides strong mechanical support to the body. It is a product of sclerotization. Next comes the lamellar organization. The chitin microfibrils, which lie parallel to each other, often form a group of layers about 20 to 250 thick. The orientation of the microfibrils differs from one lamella to other, and hence it becomes easy to recognize and count them in the sections. Observations on the locus suggest that the lamellar cuticle is produced in the night, while the non-lamellar in the day, as in the later, all microfibrils follow one and the same direction. The fully formed cuticle is this, composed of alternative lamellae and non-lamellae regions. Due to the lamellar architect, the endocuticle becomes much elastic and provides flexibility and stretching properties to the integument. The next comes the pore canal. They run throughout the thickness of the cuticle in perpendicular direction after transversing the chitin microfibrils. The pore canals are very fine structures measuring about 0.15 to 1.0 microns in diameter. They contain the cytoplasmic fine processes of the epidermal cells. In a newly synthesized cuticle, the pore canals follow the spiral course and contain the cytoplasmic filaments, while in the matured cuticle they become straight and contain cuticular substances. Distally, the pore canals are divided into the wax layer of the epicuticle after penetrating the cuticulin layer. Sometimes there is a partition beneath the site of branching, which is known as a pore plate. The pore plate separates the wax filament filled region of the pore canal from that filled with the chitin. So when we try to learn about the structure of the integument, general organization is there: epicuticle, exocuticle, mesocuticle, endocuticle, pore canals, subcuticle, epidermis, basement membrane, inocytes, dermal gland, and trichogen cells. Once again, I'm going to read it from top to bottom. Epicuticle is there, exocuticle, mesocuticle, endocuticle, pore canals, subcuticle, epidermis, basement membrane. In that basement membrane, we could see inocytes, dermal glands above that basement membrane. Then trichogen cells are also there. Some bristles are also seen. Bristles are actually, starting from this trichogen cell, which is situated in this basement membrane, and externally visible from this epicuticle layer. Okay, epicuticle in detail. Cement layer is there, wax layer, polyphenol, cuticulin layer, procuticle, and pore canals. Okay, there are about 15,000 canals per square mm in the endocuticle of Sarcophaga, while 1,200,000 per square mm in Periplaneta. The pore canals perform the function of transport of various cuticular substances from the epidermal cells onto the upper surface of the procuticle to facilitate its growth and sclerotization. Chitin protein microfibrils. The chitinous cuticle is composed of two types of molecules, the chitin and the protein chains, which are often closely associated with one another. Chitin. It is a nitrogenous polysaccharide named by Odia in 1823. It is named up of long chains of acetylated glucosamine residues, N-acetyl glucosamine. The adjacent chitin chains are held together by beta glycosidic linkages, particularly the hydrogen bonds, to form the microfibrils. The neighboring chains always run in opposite direction. The chitin microfibrils are embedded in proteinaceous cement, particularly in the region of the exocuticle. The chitin molecules are linked with the protein molecule throughout the cuticle by covalent bonds to form a glycoprotein. It forms 60% of the dry weight in endocuticle and 22% of dry weight in exocuticle in Periplaneta. It is insoluble in water, alcohols, ethers, and other organic solvents and alkalines, but soluble in concentrated mineral acids. It has about 1.4 specific gravity and 1.55 refractive index. Pore canals, a newly molded epicuticle, exocuticle, mesocuticle, endocuticle, pore canal, subcuticle, epidermis. Cement layer is there, wax layer, polyphenol layer, cuticulin layer, wax filament, pore canal, pore plate, then pore cuticle and filament layer. Okay. Biosynthesis of chitin. Chitin is synthesized by the insect epidermis. The active chitin monomers are secreted into the extracellular apoelysian spaces where they are polymerized to form chain by the enzyme chitin synthetase. Chitin synthesis in insects is controlled by 20-hydroxyecdazone and it is pH and temperature dependent chemical process. Chitin synthesis take place via a metabolic pathway to 8 or more steps converting hemolymph trehalose into chitin as follows. Trehalose turns into glucose, glucose 6-phosphate, fructose 6-phosphate, glucosamine 6-phosphate, N-acetyl glucosamine 6-phosphate, N-acetyl glucosamine 1-phosphate, then uridyl diphosphate N-acetyl glucosamine chitin. N-acetyl glucosamine, biosynthesis of chitin.